Right, good morning, everyone. I think we've got another person joining us. Hopefully you can hear me all right. Can you indicate that you can hear me either by putting your hand up or someone typing into the chat box and saying, yes, we can hear you, John. Okay, good. Well, good morning, everyone. It is just after 9 o'clock in the UK. My name is John Gogarty. I've been working with Pearson London examinations, Edexcel, whatever the version, for very many years, over 30 years. I'm currently a principal examiner for a biology specification at IGCSE, and I have been a chief examiner for IGCSE and a principal examiner for A-level. The different specifications. I don't work for the International GCSE Human Biology team other than providing training, because under the UK Ofqual regulations, if you are involved in writing examination material, in other words, writing materials for upcoming exam series, then you're not allowed to deliver training. Okay, and that's an Ofqual requirement that applies to all of the awarding bodies, not just Pearson. Okay, so good morning, everyone. Welcome to today's training, which is for new teachers of International GCSE Human Biology. Okay, so let's move on to the next slide. Okay. So what we're going to look at today, in today's agenda, we're going to look at an introduction to Edexcel International GCSE Biology. We're going to look at how you might teach it, some planning and delivery help. We're going to look at how the assessment structure is made, okay, and also look at various resources and support that's available, okay. If you look in the chat box and go backwards, I think Hansel has put in the link for the delegate material. If not, if you go to the Zoom at the bottom of the page and click the extra dot buttons at the end, it will allow you to download where it says resources. It will allow you to download today's material. But Hansel, if you could perhaps type the link into the chat box again, that might help those who are having difficulty in locating those resources. Okay. So the aims and objectives, the link is now in the chat box. The aims and objectives of today's training are to gain an overview of the specification and its content, to look at different ways that one can teach and deliver the IGCSE in human biology, to look at the assessment structure and how that assessment structure determines the best way to prepare your students for examinations, okay, and also to identify and signpost you to where support is available from Pearson Edexcel for this qualification. Okay, good. Let's move on. Okay, so a little bit about Pearson. So Pearson are the world's largest learning company, the UK's largest awarding body, depending how you work it out, and are placed well to deliver qualifications aligned to the British and international education system, and their heritage goes back over 150 years. And today they work with schools, universities, colleges, employers, training providers, to deliver world-class, globally recognised qualifications to over 3.5 million students a year. So a very big organisation. It tells you operating in over 70 countries on there. Okay, so let's move on. Okay, so let's look at this particular GCSE qualification. Like a lot of the IGCSE qualifications, it was rewritten to reflect the changes in qualifications from 2019. So from 2019 onwards, it had grades from 1 to 9, where 9 is the highest possible grade that one can achieve, and 1 is the lowest grade you can achieve that's still recognized by a certificate. In comparison to the previous GCSE system, a level 9 is above what was previously an A star. A level 8 was what was previously an A star. A level 7 was what was previously a grade A in the old system. So it is using the same system that's used in UK schools and used across all of our international GCSE qualifications. Okay. So if we look at the spec content, the spec content is described for you in the specification, which is also in the downloads and also in the delegate booklet. Okay. And it has 12 units that include modern, up-to-date and engaging science to allow students to study a range of topics within human biology. Okay. The delegate booklet shows you the list. It gives overall title for each of the 12 topics that students must study, and each topic is also divided into subcategories. The number of subtopics varies depending on the topic. The number of subtopics varies depending on the title of the topic, something that needs to be taken into account when devising a scheme of work. Okay. So the 12 units, content has been modified. It allows transition from level 2 to level 3, so from what was key stage 3 into key stage 4. It's compatible, provides in-depth information on human biology for students of surrounding professions such as medicine. The topics allow students to develop a variety of skills, including practical skills, analytical skills, problem solving, mathematical skills, we'll talk about this a little bit later, and also it allows them to develop critical thinking, and the scheme of work is linked to different critical thinking skills that Pearson highlights across all of its GCSE specifications. Okay, so let's move on. Okay, what we're going to spend a little bit of time doing, and this is in your delegate booklet, is these are the 12 topic areas, okay? And this slide is reproduced in your delegate booklet, okay? Hopefully you've all been able to look at the delegate booklet. The downloads that you have this morning contain the following documents. They contain a delegate booklet. They contain a getting started guide. They contain a copy of the sample assessment materials known as the SAMs. They contain a copy of the specification. It contains a list of the practicals. It contains a scheme of work. It contains a list of command words. It contains a document from Pearson's called Maths for Scientists, and it also contains a document called Practical Terminology in GCSE Science. So it contains 1, 2, 3, 4, 5, 6, 7, 8, 9 different documents, okay? And those resources can be downloaded from the Zoom as Hansel instructed, and also there is a link that allows you to download it, which is currently present in the chat. Okay, so what I would ask you to do, if you're able to do this, is to have a look in your delegate booklet, or if you've got a great knowledge of the specification anyway, and to work out the number of practicals, okay, that are contained within each of these 12 topic areas. So the 12 topic areas are on the screen: cells and tissues, biological molecules, movement of substances into and out of cells, bones, muscles, joints, coordination, nutrition and energy, respiration, gas exchange, internal transport, homeostasis, reproduction, heredity, and disease. So if you could, in your delegate booklet, write the number of practicals that are included in each, or if you like, you can type into the chat box the number of practicals that appear in each spec section. Now, some of them have no practicals in them, and some of them have more than one. Okay, so see if you can do that. I'll give you 3 or 4 minutes to do that, and then we'll go through the answer to that to see if you can remember how many practicals. These are the required practicals, compulsory practicals, which are illuminated in the specification. They're not the suggested practicals that appear. So have a go at writing those in your delegate booklet or type them on the screen. I say I'll go through those in a couple of minutes. So the subject content in the delegate booklet, which is pages 4 to 13, will help you with this. And to complete the table on page 14 in the booklet. So I'll give you a couple more minutes. Remember, you're not going to get practicals in every topic. You're only going to get practicals in some of them. Okay, let's have a look. So we move on. So there are no compulsory practicals associated with cells and tissues. 4 practicals associated with biological molecules. No core practicals associated with into and out of cells, bones, muscles, and joints. 2 suggested practicals associated with coordination. No associated practicals with nutrition and energy. 1 with respiration, 2 with gas exchange, non-internal transport, and 1 with homeostasis, making a total of 4 and 2 is 6, and 1 is 7, and 3 is 10. Okay, let's move on then. So there are 10 compulsory practicals, the ones we've just looked at. Okay, these can be carried out hopefully by the student. If not, they could be carried out as demonstration, depending upon your facilities that you have in your centre, or depending upon the expertise of the students that you're teaching, or their access to labs and practical resources. Okay. There are a further 7 practicals that are outlined in the specification as suggested practicals. They're not compulsory, but they could have relevance to the examination. In other words, they might be referred to in exam papers. Details of all the compulsory and suggested practicals are listed in the delegate booklet. Okay. An assessment of practical skills accounts for 20%, so between 19 and 21% of the marks available in the human biology papers. Okay. So even if the students are in a circumstance where they're unable to individually carry out the practicals, they do need to be familiar with the methods and the procedures. So they could have access to simulations or demonstrations, but they do need to know the content of the practicals because they can also be referred to in the examination. Okay. So if we look at the suggested practicals, the compulsory ones, okay. If we look at the suggested ones, there are also suggested practicals at the moment appears on the screen. What I'm asking you to do is to look at the booklet, look at the screen, and identify the spec reference of these suggested practicals. Okay. So on the screen are 1, 2, 3, 4, 5, 6, 7 suggested practicals, which are beyond the compulsory ones. See if you can identify the topic title or the spec reference, the part of the specification that these suggested practicals are from. Okay. And again, you can either type that in the chat, or if you've got your delegate booklet, write that in the delegate booklet. So add the specification reference number and the topic title to that table. And this suggested practical table is on page 15 of the booklet. I'll show you the booklet in a moment if you haven't been able to see that. And so we've got 2 people in the waiting room we might need to have a look at. Okay, what I'm going to do is to go and show you what the delegate booklet looks like in case you've not been able to access that. So I'm going to stop sharing this screen just for a moment and share a different screen with you. So please bear with me while I do this. Hopefully you can see my screen. The delegate booklet on it. It's just loading, so it takes a little while. Right, says you are sharing this screen. Okay. Hopefully. Okay. Right, so Your delegate book has your content in. So these are the 12 topic areas that we spoke about. And then with the numbers, practicals. So for example, in coordination, there are 2 practicals, 5.7 investigate the position of sensory factors such as touch, temperature, and effect on the skin, and also 5.15 investigate the range of frequency audible to the human ear. So the core practicals are written in italics within the particular specification section. The numbers we referred to earlier. Okay. And then, so let's take a little while to load. These are the other parts of the specification. Again, these have different practice in them. This. The activities that we just gave you to look at. Okay. Right, so this is, we put the number of practicals originally, and this is here, we're asking you to do the spec references for suggested practicals. To investigate different tissues, to investigate mitosis, investigate nutrient content, the food test limited to starch, protein, and glucose, investigate diffusion osmosis, investigate the speed of nerve impulse, investigate light intensity on eye, investigate the effects of antibiotics on bacteria. So the other ones that we're asking to identify the spec reference and the areas they're from. But this is what the delegate book looks like. So I wanted to show you that. That's the document that you need to download. Right, I'm going to stop showing this screen now. I'm going to turn to the PowerPoint so that we can carry on. I'll show that. So stop sharing on here. Back to sharing my PowerPoint with you. Screen loading. Okay, so hopefully now you can see PowerPoint screen. Okay, good. Okay, so go to the next slide. Right, so these suggested practicals. So obviously, investigate cells is going to be in spec reference 1.1. Recognize cell structures using a light microscope, electron microscope. Obviously, they won't be using electron microscope, but they're using pictures from that, so electron micrographs. Okay, so mitosis during cell growth and repair, so either 1.10 or 1.11. Investigate nutrient content of food. Describe a test for glucose, starch, and lipid from either 2.3 or 6.1 or 6.3. Diffusion osmosis is obviously from movement of substances into and out of living and non-living systems, so 3.1, 3.2, or 3.3. Investigate the speed of a nerve impulse is obviously from the coordination part of the spec, so that's 5.1 to 5.6. Likewise, the effect of light intensity on the eye is from coordination as well, explaining the structure and function of the eye as 5.11. And then obviously investigate the effect of antibiotics on bacteria is from the disease part, so that's 12.6. Okay, right, we've got another person to admit, I think, in the lobby, so if we could do that would be good. Okay. So I'm going to type a message to you. So hopefully, you have all been able to download the delegate booklet. Let me know if you haven't before I move on. Okay. Okay, so I'm now going to look at the mathematics content. Now, as GCSE and international GCSE examinations were reformed, maths content now makes up 10% of the total marks, okay, across all biology and human biology papers, okay? So regardless of what your students cover in the maths part of the maths GCSE, I always found as a teacher that it's much more useful to teach the mathematics as we cover that part of the course. Okay. So if I did some data on effectiveness of contraception, we would use the math skills whilst we're doing that part of the specification. Okay. So there are 5 skill areas, or 5 categories, if you like, in which the mathematical components are recorded, mathematical requirements are recorded within the human biology specification. And these are given in your delegate booklet on pages 17 to 18. We're going to look at them again on the screen. They're also given in the appendix of the specification document itself. Okay. And this gives a more detailed idea of what kind of content needs to be covered. So example might be for graphs that students would be expected to extract information from a graph and manipulate this information to form a conclusion or make a comparison Okay, so let's move on and look at an example of this. So it's important that we teach the maths within our biology lessons. Okay. So this gives us an example of the first 2 parts. HB just means human biology. Okay. So they need to be able to recognize and use numbers in decimal form. Okay. They need to be able to recognize and use numbers in standard form, and to be able to convert from decimal to standard form. Okay. So that's something we need to practice. They need to be able to use ratios, such as in genetics, fractions, percentages, powers, and roots. And students, certainly my students, were notoriously found difficulty, particularly calculating percentages, both percentage change and percentage difference, and use examples of these. And remember in the examination to always be able to show their working. So if they make an error in their calculations, they can still gain some marks from what they've done, the stages they've done. So each stage in the calculation is important. In terms of handling data, so using appropriate number of significant figures, and the appropriate number of significant figures is more important than the number of decimal places, because the significance of the figures will determine how many decimal places you need to use. So they need to understand what significant figures mean. They need to be able to understand and find the arithmetic mean, to construct and interpret bar charts, to construct and interpret frequency tables, diagrams, and histograms, to understand the principles of sampling as applied to scientific data, to understand simple probability, to understand the terms mode and median, to use a scatter graph or scatter diagram to identify a pattern or a trend between two variables. So a scatter diagram, for example, would show a correlation. And to make order of magnitude calculations, particular things like working out magnification would be important. So that's an example of the individual details of two of the components of mathematics that they need to know. Okay, let's move on a little bit and have a look at how this maths content, how we can use this maths content, or examples of using this maths content. Okay. Okay, so we're going to think about two areas. Now, you don't need to read through these particularly, you should know from teaching or experience in biology. So if we think about topic 11, which is reproduction and hereditary, and we think about topic 12, disease, think about, if we do, say, reproduction first, if we do topic 11 first, think about 3 math skills that you would use when teaching reproduction and inheritance. Okay, what skills would be useful in teaching topic 11, say? Okay. And then how might an opportunity to teach math skill 2E, principles of sampling apply scientific arrays? Let's do the first one first. So what I'd like you to do, okay, in the chat box, which is here. Okay. So give an example of a math skill and how you could use it in, let's say, genetics or reproduction. Type it, you could type that into chat box, that would be good. So have a go now. Okay, ratios, fraction, percentages, good. And how would you do that? So whereabouts within the spec content, within the teaching scheme, would you use the math skills, for example? So yes, ratios, fractions, percentages, excellent. For which particular bit do you think? Type it into a chat box so we can see would be good. Pedigrees, excellent. So you might, for example, use Punnett squares. Excellent. Genetic diagrams, pedigrees. Okay. Examples of probability, what used to be called Bayes, in other words, combining probability. What is the probability that a child will have cystic fibrosis and be male? So combining probabilities even. Okay. What's the probability of a second child having haemophilia? Okay. Any other examples you could think of? How about the relationship between probability and gamete formation? Have a go typing some other examples. What about from topic 12, disease? See if you can think and type into a chat box for me some examples of using math skills from either reproduction or disease. Okay, for either of those. Type them into me and we'll have a look and see what you come up with. That would be really good. Thank you. Okay, any others? So an example that I could consider, or I considered, is if you were looking at the effectiveness of contraceptive methods, what maths could you use to compare them? Okay. So what math skills? Oops, sorry, me. Computer keyboard's going mad. What math skills could you use to compare the efficacy of contraceptive methods, for example? What might you do? What could we do? Type your answer into the chat box. What could you do? What statistics might you compare? How would you compare, do you think? Okay. Well, the way they normally compare contraceptive methods is the number of pregnancies per woman per year or per 100 women per year, whatever. So, and they can measure the efficiency of that. So, for example, they might say that, you know, in 100 women using an oral contraceptive pill, you might expect 3 to 4 pregnant every year. So that will be a 97% efficacy rate. So that kind of thing. So we need to be aware of what to do. If you're looking at disease, you might be looking at red blood cell counts. So the students might need to know, okay, in terms of maybe how a hemocytometer works, or in terms of the scaling up in terms of dilution, the number of red blood cells per cm cubed of blood, and they would express that in standard form. Okay, so they need to know how to use standard form. Okay, so that's a kind of thing that we might expect them to be able to do. So what we need to do as teachers is, whilst we're teaching these topics, to ensure that when we look at scientific data, comparing contraceptive methods, when we look at the effect of haemophilia, for example, on red blood cell count, or the effect of drug treatments on white blood cell counts, that we are using examples that make the students familiar with the math skills are likely to be examined upon, such as standard form, such as dilution, such as probability. Okay, so those math skills need to be covered as we deliver the relevant part of the specification. Anytime you have an opportunity to reinforce those math skills, then we should try and do that, because that helps the students. Okay, good. Okay, let's move on. So it's an example of math skills we could use. The question 2 about delivery of a less conclusive analysis and manipulative way. Okay, the number that had successful pregnancy, so this is the opposite to what I considered. I considered perhaps using it for contraception. They were using it as a IVF treatment or trying to help people become pregnant. Then you could live using the number of successes in pregnancy. Okay, or data could be conclude the quality of life has improved in cystic fibrosis patients that receive gene therapy compared to those not receiving gene therapy. So again, these are examples of where we can use math skills to support our delivery of the spec content. Okay, good. Let me move on. So what we're now going to look at is how you can plan and teach delivery of this particular course. Okay. Okay, let's move on. Right, so when you're planning to teach the specification, you need to obviously know the specification and where there are opportunities to teach subtopics together, okay, which may save time. Okay, so for teachers of experience of teaching human biology, they might understand the changes to the qualifications from the previous version. To incorporate key skills, we've just spoken about this, haven't we? About maths, about practical work, about scientific literacy, particularly, I think, using sufficient detail and using precise language, okay? To look at, for example, extended writing for answering those questions that have, say, 3 or 4 marks, the explain questions, the describe or the discuss questions. Practice at comprehension, because comprehension question comes up on paper 2. To ensure that all compulsory practicals are accessed by and accessible to the students, and to look at how you might include some of the suggested practicals in your scheme of work. What you also need to do, excuse me, is to think about how you might teach or decide upon a teaching order. Okay. When we're thinking about how topics link together, those students that are able to secure higher grades are often those that are able to apply their scientific understanding and knowledge to link together situations, okay, across different parts of the specification. Okay. And in the downloads, in the Getting Started guide, which we can download, okay, it shows you those parts of the specification that have changed. In other words, where there is new material. We've also looked at the type of opportunities where different types of mathematics can be included in your planning. Okay. Students, remember, can be tested on any of the math skills. They can be tested on any of the practicals. Okay. And they can, particularly the required or core practicals, but also it helps them if they have a knowledge of the suggested practicals we spoke about. Now, the order that you teach the specification in is not set in stone. Okay, you might want to change the order depending upon the circumstances of your centre. Subtopics can be combined, and then that can help us determine what the best order is. And this is down to individual centres to decide. So simple science is demonstrated in topic 1, so cells and tissues, and subtopics 1.1, also in topic 3. Movement of substances in and out of cells. These two topics could be combined and taught at the start before moving on to more complex topics such as DNA and protein synthesis. Okay. There are schemes of work that you can download from the same place that you downloaded your delegate booklet. So schemes of work are available. Okay. Let's have a look at the next activity. Okay, so there are some crossover topics, as we said, in different parts of the specification. And these could be taught together. So if we look at topic 2 and topic 6, which are in the specification booklet, sorry, in the delegate, but I'll show you these in a moment. Which topics could be taught together? So we're looking at topic 2. And we're looking at topic 6. I'm going to go into a booklet to show you these in a moment. How could these be taught together? Okay. And what opportunities, for question 2, what opportunities are there to develop maths and extended writing within topic 2 and topic 6? So what I'm going to do, in case you're not being able to download the delegate booklet, is I'm going to look at these topics from the delegate booklet with you. Okay, so I'm going to stop sharing this screen for a moment. I'm going to go back in and look at the delegate booklet. We're going to look at topics 2 and 6, and then we're going to try and identify where some of the content can be taught together from topic 2 and topic 6. So I'm going to stop sharing this screen for a moment. Okay. I'm going to go back in to some of my here. Find the delegate booklet. Find it. Um, so this will work. Right. So it's going to once we load. Load it. So right. So looking at topic 2 and topic 6. Okay. Sorry, this is slow. The top. Right. So topic 2 and topic 6. So topic 2. Right. So topic 6 is nutrition and energy. Okay, which we've got there, whoops. And topic 2, just going to share it now, silly. Topic 2 is biological molecules. So we're looking for overlap between biological molecules and nutrition and energy. Okay, so have a look through those and see what aspects of 2 and 6 you could teach together. Okay. I'm going to go back to the PowerPoint. So there's 2 and 6. Go back to now. 6 was nutrition and energy, wasn't it? There's nutrition and energy. Okay. So 2 and 6. I'm going to go back to the PowerPoint. So which content from 2 and 6 could be taught together? Okay. And what I'd like you to do is to type in excellent, good. So there's some nice suggestions there. Have a go at typing in where you think there's overlap between topics 2 and 6. Please type in the box. And we're right, type in here, please. So some excellent examples, relationship between enzymes, biological molecules and nutrition. What else could you do? What else overlaps between 2 and 6? I'll give you a couple of minutes to type some in the box for me, please. That would be really good. Thank you. Any examples of overlap between nutrition and biological molecules? There should be. Have a go. Type in the box for me, please. Thank you very much. Anyone else? Type in the box for me, please. Thank you. Good. Excellent. Any others? Okay. Let's move on. There's some examples there, compulsory practicals together, math skills that overlap, chemical breakdown, which we mentioned, carbohydrates, protein, lipids in the digestive system. So a relationship between enzymes as molecules, the enzyme content, enzymes as proteins, and also their role within the digestive system. So there's lots of overlap between biological molecules and their functioning, and their role, therefore, in nutrition and energy. Okay, good. Let's move. So here's some other examples. This is a planner. Let's check down a little bit. So this shows you an example of planner. This 2-year course planner is available in Getting Started Guide. Getting Started Guide is one of the downloads that you have. Okay. It shows a 2-year plan. It can be found. It not only gives the idea of the amount of time that could be spent on each topic, but also summarizes the topics that need to be covered. This could be used for delivery of a course or could be adapted to better suit the needs of individual establishments. I think if I remember, the Getting Started guides exist as Word documents, so you can edit them. Sometimes things work are downloadable as PDFs, and there are problems editing PDFs, but there are not so many problems editing Word documents. So this is suggestion, number of weeks, suggestion of order of topics, how long it might take, and subtopics covered. Okay, so this is a course planner. It's not lesson plans. Okay, but it gives you an idea of maybe how much time to spend on each if you are new to delivering this. Okay. Do you teach the course? Is a question, and hopefully we'll have a bit of more interaction through the chat. Do many of you teach this course over 2 years, or do you teach it over 3 years? Can you type your answer in the chat box? 2 or 3-year plan. Because some schools teach GCSEs over 2 years, some teach them over 3 years. Have a look. It'd be interesting for me to know that. Do you teach 2 years and 3 months? That's interesting. Good. Some people teach them over 3 years, or they might start, as the delegate kindly wrote there for me, they might start towards the end of, say, year 9, the 3rd year, and do the rest in years 4 and 5, years 10 and 11. Anybody else tell us whether they teach them over 2 years or 3 years? Type your answer in the chat so I can have a look. That would be great. Thank you very much. Type in the chat. The chat is at the bottom of there or the top, and it says, well, little chat bubble is comic bubble. 2 years, okay. 1.5 years, that's very quick. Okay. Hopefully you have more lessons per week. Okay. Do many of your students also take biology? Okay. So do your centres do both biology and human biology? I'm going to type that here. Do your and then we'll move on, because obviously there's going to be some overlap between biology and human biology, but is it different teachers that teach it? Be interesting to know. Okay, yeah, most people. Good, good. Okay, so there are suggestions there that of a plan and how you can alter those plans depending on the individual demands of your centres. Okay, let's move on. Thank you very much for that information. That's very useful. Okay. Right. So there are different teaching strategies that we said we would consider. Okay, and it gives you some examples here. There are many online resources for teaching strategies. This is often a matter of individual taste as to how you're going to teach things. in my experience in terms of teaching strategies, obviously it helps if you are able to have a varied approach, okay, so that you can have different teaching episodes in each lesson. So another role I have, but not for Pearson, for another university within the UK, is that I visit student teachers on teaching is a variety of episodes within each lesson. So you wouldn't spend the whole lesson doing a PowerPoint like we're doing here. You wouldn't spend a whole lesson doing a long practical. You would do different episodes within the lesson, and you divide the lesson up into those different stages, and you manage the transition from one stage to another. So there are lots of online resources that you can use. Okay, there are also templates available for students to construct, for example, making a model of DNA. Gives you an example of that. I've seen these paper ones they can cut out that form a helix. I've seen them done with pipe cleaners. I've seen them done with post-it notes. So lots of different ways of teaching. Some students use mind maps. It's a good idea to use sample assessment material or past papers, and I think it's a good idea to do them within the lesson. So when I first started teaching a very long time ago, you would teach the lesson and then you'd have an exam or a test or a quiz somewhat later than that. What tends to happen now, I think, is better practice, and I think the students find it more helpful, is to integrate your assessment for learning, so your formative assessment rather than your summative assessment, but do that throughout the lesson. So you would teach about osmosis, you do definitions, you discuss it, you give some examples from different parts of your specification, and then you'd have some exam questions upon that within the lesson. Okay, so where it says using past papers, examiner reports are helpful, I would do that in the lesson rather than just saving exam questions for an examination situation or a mock exam or a quiz. Okay, and key and practical terminology can be reinforced. They're suggesting crosswords or word searches, and there's lots of material out there that enables you to make your lessons more varied. Okay, I think the more we can integrate the assessment, the formative assessment, into the lessons, the more likely students are to be able to remember and associate their knowledge and understanding with its application through exam questions. So my view would be to use exam material or practical material or stimulus material within the lesson to give a varied approach. Okay. So what Pearson would like you to do, what I'd like you to do, is to think of a teaching strategy that you've used in a lesson, and one that worked well. I'm not terribly interested in one that didn't work well. Why would you want to say I did this and it didn't work? So think of an example, maybe from digestion, maybe from movement in and out of cells, maybe from another area of the specification. And if you could be kind enough to share that example with the colleagues this morning and type that into the chat. So if you could type an example of an unusual strategy, maybe. That worked, and then we can have a look at some of these. So type them in the, you'd be kind enough to type into the chat. That would be really good and really helpful. Thank you. And I'll think of some examples myself. So this could be modeling. It could be a model to show digestion. It could be a model to show osmosis or dialysis or whatever. The relationship between temperature and enzyme action, whatever. It'd be nice if someone could type an example into the chat rather than just me wittering away to you. That's a nice example. Good. Excellent. Any others? What else have we done? So someone other than me would be good typing these. What else we could do could be done? Um, any others? Excellent. That's really good. Thank you. Any more? See if you can come up with one each. That'd be really good. I'm going to get a glass of water. I'll be back in a bit. Okay. One of the things I think works quite well is um, my typing's pretty bad. Okay, so one of the things I've done, I've noticed across both biology and human biology papers, is often students don't really understand the relationship between concentration and osmosis. So they'll talk about concentrated solutions, and then they'll talk about concentration of water. And the concept of concentration of water is not usually very helpful. So we would describe osmosis as the movement of water from a dilute to a more concentrated solution. Okay, or you could talk about water if you want, you don't have to. But they need to understand that a solution has two components. It has a solute and it has a solution. And I think I used like different concentrations of either blackcurrant squash or orange squash or whatever, and show them how dilution works, and they can understand the relationship between the solution and the solute concentration, where the solute would be the blackcurrant or the concentrated, and then you add water to it, it dilutes it. I think that's a good thing, because they find that quite hard to understand, and sometimes textbooks aren't very easy either. I've seen people using visking tubing to make balloons or sausages and putting them in different solutions. To make model cells. And then you can put these in different solutions and look at the changes in how flaccid they are and how to, etc. So there's lots of different things we can do with model cells. I've seen students make model cells with plasticine, with Play-Doh, whatever, to show a 3D structure in cells with a nucleus, etc., etc. So lots of different examples. Okay, and thank you very much to those of you who are kind enough to give some examples for us to look at. So thank you very much for that. Let's move on. Okay, so this gives you an idea of resources and support that are available. Okay, so we've got Getting Started Guide, which is in the download today, as is a scheme of work, skills mapping, sample assessment materials, as are example marked responses. I think these are downloadable from the Pearson website. Okay, and results support. So you've got subject advisor, which used to be called Ask the Expert. Well, you can push and they will send your question to a member of the senior examining team for human biology who will reply to you. Okay. You've got past papers, you've got exam wizard, which we'll talk about later. You've got mark schemes, you've got results plus, which we'll talk about later. You've got access to scripts, which we'll talk about later. And also you've got page resources and other textbooks that you can buy. You don't need, and we will say this, you don't need to purchase page resources in order to be able to teach the specification. It's important to say that, okay? So that's a support table, and I particularly encourage you to use Subject Advisor, which is actually the expert, because they are very good at asking questions. And you can ask a question. If you don't understand the answer, you can go back and say, I don't understand the answer. Okay. What we can't do is rewrite mark schemes. So you might say, oh, I didn't like the marking in January 19 or November 19 paper. Don't understand why that didn't get a mark. You don't need to agree with the mark schemes. I've marked papers, and my students have sat papers where I don't necessarily agree with the mark scheme. But what you need to do is to understand the mark scheme and how it's been applied. You don't need to agree with it. You might think this shouldn't get a mark or this should get a mark. That depends on the chief examiners and who set the paper. But you need to understand how the mark scheme works so that you can be better informed with your teaching in future. So it's about understanding the mark scheme. That's really important. Okay. And a glance of the different support available. Right, we're going to say a little bit now about the assessment structure. Okay. A little bit about how these papers are structured, which we're going to do. I'll just get rid of that on the screen. Okay, so let's move on. Okay, we've done that. Resources and support. Right, let's move on. Okay. Okay, next one. Right, the structure of the assessments. Okay. These next few slides give us an idea of the overview and how to prepare students for the assessment. Right, so these changed from January 20, and the new structure is not in place, so relatively new. Okay, so there are the two papers. Okay, paper 1 and paper 2. They both have 90 marks, and they both make up 50% of the qualification. Okay. Each paper contains questions that cover a broad range of subtopics. Okay. So any of the topics can appear on paper 1, any of the topics can appear on paper 2. Okay, good. And in no sense does paper 1 do the first 5 and paper 2 do the second 5. Okay, so both papers cover all of the content. Okay. It is what's called a terminal assessment. In other words, it's not a modular assessment. All the candidates take both papers at the end of the course. So the end of a 2-year course or the end of a 3-year course, or however long you spend preparing them. And the papers, I think, are available in November and in May, November and June. Okay, so summer series and autumn series. Okay, this is a summary of what I've just been through. So it tells you linear assessment, 2 papers at the end of the course, 100% external assessment, there's no coursework. There is a single tier of entry. This is important. So this means that the whole range of grades, from the lowest grade that gets a certificate, so grade 1, right through to the best students getting a grade 9, are examined through the same papers. So those papers cover the whole range from grade 9 down to grade 1. All components, maths and practical, are tested to meet the assessment objectives. And there are a mixture of questions, including comprehension and extended writing. Okay. And I think the comprehension is on paper 2 only, not on paper 1. Let's move on then. Right, here we go, sorry. Right, so each paper consists of a mixture of different style questions, calculations, and remember what I said earlier about the importance of showing your working in calculations, that you maximize the number of marks that are available. Remember to, I think a good tip is to encourage students to practice rounding. So often they'll, if the answer's 23.66 recurring, then if you're doing that to one decimal place, that would be 23.7, okay? Or if you do two decimal places, that would be 23.67. Sometimes they just put 23.6, and that can cost them marks. So practicing rounding as well as practicing significant figures is an important skill. There'll be multi-choice questions. There are short answer questions, comprehension, we said, and extended open response questions. Both papers include similar styles of questions, although paper 2 includes a comprehension with questions based on the content, which is what I've just said. The maximum number of each style of question is set to ensure a fair balance all question styles and incorporate the use of command words. And remember, command words are featured within the specification, the appendix at the back of the specification, and I think in your delegate booklet as well, there's a list of command words. And the command words are important because command words inform the student what they need to do. So example of command words would be describe and explain, state, give, calculate, okay? And we'll talk about command words later on. I think we talk about them in next week's training, the second part, when we do some work looking at student responses as well. Okay, so the questions across the two papers are devised to ensure that all the assessment objectives A1, A01, A02, A03 are examined. We'll say about that in a minute. That they use a range of command words and a range of style of questions, as well as covering the spec content. So, in the assessment of practical skills, which is A03, students may be tested on their ability to solve problems, apply scientific knowledge, devise and plan investigations, evaluate data and methods, draw conclusions, analyze and interpret data, communicate findings from experiment, describe safe and appropriate practical techniques, identify outliers and suggest improvements to methods. So all of these are about A03. Okay, so these are all associated with A03, which is application of scientific skills to practical and data from experiments. Okay, that's the importance of A03. Okay, let's move on. So they need to be able to do these. So I would practice these. Devising and planning investigations would be QORMs, for example. We'll talk a little bit about that later. Okay, let's move on. So here we are, the ones we just looked at with experimental skills. So as well as the spec content we referred to, the specification and the exam papers are designed to ensure that each of A01, knowledge and understanding, A02, application of knowledge and understanding, and experimental skills analysis and evaluation, the marks are divided into 40, 40, and 20 in terms of percentage, but also the number of marks, so 36, 36, and 18, okay, across each paper. So your teaching needs to reflect this. You can't just expect students to learn and understand the spec content without being able to apply it and analyze it. Okay. Equally, and my students have said this, if you've got 60% of your marks on application and analysis, it doesn't mean you don't need to know content. You still need to remember content. You still need to know the difference between an artery and a vein. You still need to know how the heart works. You still need to know how osmosis works. You still need to know how diseases are inherited. You still need to know the difference between red cells and white cells. You still need to know about sewage treatment. You still need to know about all the contents and contraceptive methods and whatever it is. So you can only analyze and evaluate. You can only get marks for AO2 to AO3 if you have that AO1 knowledge, secure knowledge to apply. You can't apply knowledge if you don't have the knowledge in the first place. Okay. So in terms of preparing your students to take these exams, the examiner report is helpful. Okay, it helps students and it helps you. And it shows the areas and misconceptions, etc., where students have done well or less well in previous examinations. You need to familiarize students with key terminology and key practical terminology, which is used in that they should use in their responses. And this is about being familiar with the right language and also being precise. If they mean an artery, say an artery. If they mean a capillary, say a capillary. Don't use blood vessels when it's about a particular type of blood vessel. Make use of the sample assessment material, which you can download today, also on the website, and past papers that can be used alongside grade boundary information. Okay. What I would say is if you're using past exam papers or you're using material from Exam Wizard, which is an online resource that allows you to, as a registered Pearson Centre, it allows you to develop and build your own exam papers using items from previous papers with accompanying correct mark schemes and also examiners' reports, is to make sure that those AO1, AO2 and AO3, as well as the spec content, is reflected in those things that you use. Okay. And then it says ensure that students have adequate opportunity to access practical material as well, able to name, label and draw diagrams, etc. Okay, so they practice all of these skills. Okay. Practical terminology and indeed the lists of practicals is also downloadable from today. So in the link that you were sent or in the resources button from the more of the Zoom has those documents. The documents are also available from the qualification page for human biology from the Pearson website. And it gives the practicals, it gives explains the core practicals, and it also gives some examples of exam questions where knowledge of those practicals was required. So that's a really good document, the guide to human biology practicals, really good document. So I encourage you to use that with your students. It will certainly help. Okay, moving on. So help prepare students in writing longer prose answers that can carry a large number of marks. It may help initially give students a mark scheme and help them to mark each other's. So I'm sure these days they're all used to doing self-assessment or peer assessment, use quizzes and tests to help revise and revisit prior learning. Most lessons now start with a review of past learning that provides a transition from what you learned last week to what you're learning today. Okay, and development of revision flashcards, etc. I'm sure you, as experienced teachers, you know all about these ideas as a way of preparing students so that they do their best. Okay, I think certainly revisiting, and we spoke about the links between, say, unit the content in 2 and content in 6. When you're teaching topic 6, you can refer and based on the assumed knowledge of topic 2. And likewise, when you're looking at more applied examples, they still need to know the AO1 that corresponds to those AO2 features. Okay. Practice testing students on content Practicing application type questions, discuss questions, applies questions, analyze questions is useful exam practice as you get towards the end of a course. The start of a course, perhaps more simple, straightforward AO1, AO2 questions. As you move deeper into the course, into the second year, more explanation, analysis, more AO2, more AO3, as the students are getting better at human biology and looking at themes that go across all of those content areas. Okay, we've done that. Okay, where is additional support available? We looked at this already, but here's another summary of it to help you. So getting started guide, we've got training event that this one, study advisor support we've got, community forums, you can sign up to, schemes of work are available, skills and mappings available, sample assessment materials available, examiner reports are available. I would share the examiner reports and discuss them with students. Don't forget to give students their own copy of the specification. When I taught in a girls' school in London, the students used to sit in front of me with the spec, A-level spec in front of them to make sure I didn't get anything wrong. Okay, I'd rather know at the time than later, if that helps you. Exam Wizard, we're going to say a little bit about in a moment. Exam Wizard is really useful. It allows you to compare year on year. Results Plus for mock exam analysis, Results Plus useful. I think the thing covers that, but access to scripts is useful. It allows you to target if you want students to, I think the deadline's just passed, but if you wanted to do remarks of exams, access to scripts is really useful because a student might come out the exam and saying, oh, I did really well, I got the genetic question right, I don't understand why I didn't get a grade 9. And then with access to scripts, you can go in and look at their response and say, well, the reason you didn't get a grade 9 is your gametes were wrong or you didn't put the right phenotypes in. So it allows you and the student to make a more informed judgment before deciding whether to go for a view of marking or not. So that's the benefit of access to scripts, which I think is really useful. Okay, let's move on. Right, this shows you, this is very interesting, this shows you the a slide of what the Pearson website looks like, which is really useful. So it shows you on here where you can find all the support material. Okay, so you've got, if you log into International Human Biology, this is what your screen looks like. And then you've got course materials. And then if you click on that, it goes down and you've got specification, notice, sample assessment materials, and you can filter by those. And then you've got published resources and you've got training. So all of these things is your starting point. So where it says specification of sample materials, you click on that, it shows you those 4. If you click on exam materials, it would show you past papers. Teaching and learning materials also would be more information for you. And if you click on each of those, it expands, which is what that screen is showing you. So that's a really good illustration of what is helpful. And you've got getting started guide at the bottom. You've got science guide, welcome back on useful documents there as well, which you can click on from the website. Results Plus is really useful. It tells you on there how Results Plus works. It's an online resource, supports teaching and learning, allowing a comprehensive insight into students' progress. It identifies gaps in knowledge, as well as skill areas that need further improvement. It's good for looking at your centre performance compared to other centres in the same region or across regions. If you've got different classes within the same establishment, if you're teaching a human biology class and Mrs. Jones is teaching a human biology class, it allows you to compare performance. It shows you on which parts of the specification your students have done well or less well. It shows you on which type of question your students did well and less well, and it informs reflection upon the effectiveness of your teaching. So if my class in human biology are doing very well on the disease part, on spec content 12, whatever it is, but Mrs. Jones' class are doing better, I might say, well, what is it you're doing with your class that's making them achieve better than mine? Okay. If it is that your students aren't doing very well on the comprehension questions on paper 2, it'll show you that. And then you can think, well, what do I need to do to improve that? Okay. So it allows you to reflect on student performance. It allows you to reflect on where your students are doing really well compared to other students in the region or across regions. And likewise, it allows you to reflect upon your teaching and where you might look to improve the understanding, improve student understanding in those areas where you're doing less well. So, and it shows you how to sign up on that, which is really useful. So Results Plus is a very useful and valuable tool, okay, which we spoke about. Let's move on now to the next one. It shows you there we are, more about Results Plus on there. So it takes exams found, the exams aligned, and then you can look at performance afterwards. It allows you, let me move my chat box down a bit. It allows you to analyze performance, as we said, across different spec content and question types as well. Okay. You can register for. It's a free tool for teachers, helps you make quick homework assignments, topic tests, and mock exams. Questions are you can filter against unit, topic, and assessment, or choose a whole past paper. It has existing mark schemes. Mark schemes don't necessarily have accurate marking, but it gives you existing mark schemes, so you can try and apply the mark scheme the way the examiners will have done. It gives you the examiner report for each item that you might have, which is also really useful. It has recent exam content available quickly. We try and do it. I don't know if the last summers are on there yet, but they will be soon. And you can use results, understand. So it allows you to do assessment for learning. It allows you to look at where they need extra help if they don't understand movement in and out of cells. You can spend more time on that, genetics and Punnett squares and probability really well. Then you perhaps don't need to visit that as a strategy in future. So it does inform your teaching really well. So I really like Exam Wizard. I use it lots. It also allows you to store. So it gives you a login, and you can store your, having built a paper, looking at, say, disease or looking at topic 2, whatever it is, biological molecules. You can build a paper on biological molecules and use it again next year. And then you can look at how this year's cohort have done on molecules compared to last year's. So it's very useful in comparing year-on-year performance, your performance and the students' performance. Access to scripts is really useful. So it allows teachers to access the marked papers, we'll say immediately after results day. And it offers transparent approach. It provides better understanding of marking before EARs or ROMs are done. And it teaching and comparing other cohorts. Okay, and that's useful. So post results are a useful addition. Okay, what it means is when we do ROMs, when we do review of marking, we tend to only get reviews of marking where the students are close to, when I say close to, I mean above, sorry, just below a grade boundary. So we're more likely, if there is a review of marking that increases the marks, it means that more of the reviews of marking, more of the inquiry about results that we look at are likely or more likely to produce a marked change because the centre's already carefully looked at the student responses, rather than just saying, oh, well, we'll get them all remarked, without the teachers looking at the student performance. Okay. You can also get curriculum matched student books with active books for human biology. As I say, you don't need to use these. It's not a requirement that you use them, but some centres find these useful. And there it is. There's a picture of it. International Human. There's a student book. It shows you that. Bradford and Potter. It's been around for a while. And that's the book that lots of centres use to help support their teaching. As I say, it's not a requirement, but some centres find it useful. What I will say is that your dedicated subject advisor is someone called Tim Lawrence, and there he is. Okay, there's his contact, Teaching Science at Pearson. You can send questions to him. He may or may not be able to answer them himself. If he's not able to answer them himself, he can often, or he will, send those questions to members of the senior examining team, which would be the human biology senior team, who then work through what's called Ask the Expert to respond to your inquiry. And as I say, what you need to ensure that you do is that you understand the response, even if you don't necessarily agree with it. Okay, so there's Tim's contact details, and they also appear on the website that we looked at earlier, the qualification page. So that's very useful. Right, it is now just about 10 minutes before 11 o'clock. Thank you very much for taking part in today's training. Thank you for those of you who responded to the chat and who shared your experience in terms of teaching strategies, also shared your experience in terms of helping answer my questions. If you have any questions, please type them into the chat, okay? And I will endeavour to answer them. If I can't answer them, then I can direct you to probably Mr. Lawrence or Ask the Expert at Pearson or customer support, who may be able to provide that answer for you, okay? I'm not, as I said, directly involved in any of the marking processes or setting the papers for human biology, because if I was, I wouldn't be able to do this training. Okay. I am involved in writing questions for biology and for marking biology as a senior examiner, just not GCSE Human Biology. So thank you very much for your engagement this morning. Please type any questions here, and I will try and answer them. We've got training, I think it's a week's time, exactly. I think it's exactly a week's time, I'm not entirely certain. I think it's at the same time where we'll carry on and look, I think, a little bit more detail at some of the assessment and some student answers and using mark schemes, etc., from within human biology. If you haven't got any questions that you want to ask now, or you haven't thought of any, please write them down yourself and bring them with you to next time's training, okay? And if you have a question that I'm unable to answer this morning, I might be able to go off and research the question and come back with an answer next time, okay? But please type questions if you have them into the chat box, and I will endeavour to answer them. If you haven't got any questions, can I just thank you once again for your participation this morning. And I will see you next week.